Photosynthesis occurs in the chloroplast of a cell. A chloroplast is made up of stacks of thylakoids known as grana. The stroma is a fluid-filled area of the chloroplast in which the Calvin cycle occurs. Within the thylakoid membrane there are pigments which absorb all colors of light except green. This light absorption is then used in photosynthesis. To start the light-dependent reactions, we look in the thylakoid membrane. In photosystem 2, a ray of sunlight strikes down and triggers the reaction. The energy bounces from pigments until it arrives at P680, which then donates an electron, which the electron snatcher takes away to the electron transport chain. In photolysis, a ray of sunlight hits a water molecule, separating it into 2 protons, 2 electrons, and 1 half O2. This releases an electron, which then replenishes the electrons in photosystem 2. The protons formed help contribute to the proton gradient, while the oxygen is emitted as waste. Still in the thylakoid membrane, we have the electron transport chain. The electrons move from photosystem 2 through the electron transport chain, which takes them to photosystem 1. In photosystem 1, another ray of sunlight re-excites the electron, which is once again passed between pigments until it's snatched up by the NADP+ reductase. The electron is used by NADP+ reductase to reduce NADP+ into NADPH, which is then sent off to the Calvin cycle. In ATP synthesis, also known as chemiosmosis, the proton moves down the gradient from the lumen through ATP synthase. The energy from the diffusion is then used to turn ADP and inorganic phosphorus into ATP, which is then sent off to fuel other cellular processes. The Calvin cycle in the stroma of the chloroplast is the light-independent phase of photosynthesis, consisting of 3 parts: carbon fixation, reduction, and regeneration of RUBP. The carbon fixation begins with rubisco and an intermediate taking in CO2 to convert it into a usable form of carbon. ATP then donates a phosphate group into the intermediate carbon, forming ADP as a byproduct. NADPH then reduces the intermediate, turning into NADP+. From this, the first G3P is produced and is represented by this pink pool noodle. The intermediate is now 5G3P, which is rearranged into RUBP using ATP. After 6 rounds, 2 G3P are formed, and they go on to make 1 molecule of glucose. Cellular respiration begins with glycolysis, which is split up into 2 phases: energy investment and energy payoff, the precursor to both aerobic and anaerobic respiration. In energy investment, the glucose molecule is taken from the stroma into the cytoplasm. 2 ATP are hydrolyzed, donating a phosphate group to glucose and becoming ADP. Energy investment comes energy payoff, in which 4 more ADP join in the reaction, turning into 4 ATP and resulting in a net payoff of 2 ATP. Then 2 NAD+ are reduced, turning into 2 NADH. The ATP is used for other cellular processes. The 2 NADH move on to the ECC, and the 2 new pyruvates move on to be oxidized in the next step. Pyruvate oxidation is the next step and is also known as the linking step, which takes place in the mitochondrial matrix. Each pyruvate molecule reduces 1 molecule of NAD+ to NADH. Then the pyruvate gives up a carbon group, which is expelled as CO2 as a waste product, turning the pyruvate into an acetyl group, which is then joined with coenzyme A, becoming acetyl-CoA. Acetyl-CoA will then transport into the matrix of the mitochondria to be part of the Krebs cycle. This happens twice, once for each pyruvate molecule. The Krebs cycle, or the citric acid cycle, happens in the matrix of the mitochondria as part of aerobic respiration. It begins with acetyl-CoA dropping off its acetyl group, where it joins the 4-carbon intermediate. The acetyl group produces 3 NAD+ and a 3 NADH. This doesn't really happen all at once as depicted, but let's pretend it does. The acetyl then reduces FAD+ and a FADH2 in a series of redox reactions. Finally, the acetyl phosphorylates ADP and inorganic phosphorus into ATP. The group loses its 2 carbons during this process, expelling 2 molecules of CO2. The 4-carbon intermediate, known as oxaloacetate, is regenerated and the cycle can repeat as pictured. We've entered the home stretch where the majority of ATP will be created. These last steps occur on the inner mitochondrial membrane. As electrons move, the protein complexes pump H+ ions into the inner membrane space from the matrix, creating a gradient to be used later. High-energy electrons from glycolysis in the Krebs cycle enter the electron transport chain, slowly losing energy as they progress. The first protein complex involves the oxidation of NADH into NAD+. The second protein complex involves the oxidation of FADH2 into FAD+. The final complex snatches up the electron after its energy has been depleted, combining it with hydrogen and oxygen to make water. Chemiosmosis is the part of oxidative phosphorylation where the ATP is formed. Using the proton gradient, a proton is pushed through the ATP synthase from the inner membrane space into the matrix using the proton motive force. The energy of its diffusion is used to form ATP. Thank you.